Very good evening, everyone. Welcome you all to the live practice of the environmental biotechnology course. So these are the concepts to be covered this week. Mostly we'll be discussing about the metagenomic approaches in the context of microbial community analysis, and we'll also be learning about bioremediation, like basics of bioremediation. So what does the term metagenomics refer to? So metagenomics is nothing but the study of microbial communities with the help of genomic entities like DNA. So basically, if a group of microorganisms is present in our environment, so we'll not be know whether all those microbes can be grown in the laboratory scale settings. So what we can do is simply we can extract the genomes of the microbes and we can identify them by sequencing methods. So this is how metagenomics is working. And basically, metagenomic approaches are augmented with computational tools. Usually, we'll be checking the similarity of the identified sequence with the already available sequence in the repository. So this is how metagenomics is working. So as a research field, how can we describe metagenomics? So as a research field, how can we describe the metagenomics? So usually the term meta means it is nothing but it is a kind of science which helps in the understanding the biology at the aggregate level. In the sense, so in metagenomics we will not be studying only one microorganism. So we'll take into consideration the entire microbial community. So as we are understanding the biology at the aggregate level, which means the entire microbial community, the term meta is being used. So the term metagenomics refers to studying the genomics of the entire microbial community present at a single place. So usually with the help of metagenomics, we'll be studying how the microbial genes are getting influenced by the surroundings and the interaction between the among the different microbial species. So this is the basics of metagenomics. So why we should rely on metagenomics? So usually in metagenomics, we won't be sequencing the genomes, not sequencing, like we won't be cloning the genomes. So in the previous techniques and all, we have studied a lot of techniques previously. So in those techniques, what we'll do will be studying the phylotypes and functional markers, and based on that we'll be identifying the organisms. But in the case of metagenomics, we'll be directly sequencing the DNA. We won't be using any PCR based techniques in order to amplify the genome. But what we will be doing is we'll be directly sequencing the genome, the metagenomics. So basically what we will be doing is we'll be constructing longer sequences known as contigs. So with the help of bioinformatic tools, we'll be constructing longer sequences called contigs. So with this contigs, we'll be sequentially arranging the contigs in order to get the entire genome. So this is done by the process called assembly, in which we will be sequentially arranging the DNA sequence based on the overlapping sequence of the DNA fragments. So for say if we have two DNA fragments, and at the end if the DNA fragments are having and at the extreme ends, if the DNA fragments are having overlapping sequences, they can then we can arrange those two sequences together. So this is how assembly is being done. So in this way we can arrange the closely related genomes together, and with by arranging these closely related genomes together, we can identify the similarity between different species. So this is the basic application of metagenomics. So what are the outcomes we will get out of metagenomics? The first and foremost outcome is the community composition. We can identify what are the organisms present in a microbial community with the help of metagenomics. Then we can identify the metabolic potential of different organisms by studying the genes and pathways. So what we can identify, firstly we can identify the community composition, then we can realize the metabolic potential of different species present in the microbial community. So after that, what we can do, after identifying the metabolic potential, we can select specific RNA to realize the metabolic potential. So with the help of that, we can say which gene is responsible for a particular function. So that is one outcome. Then we can construct a network and models, like models of a microbial community. So this can be done with the help of multiple samplings and data sets. So with increasing number of samplings, the precision of the models can be increased, and we can easily predict what can happen in a microbial community. So these are the basic outcomes of metagenomics. So what are the advantages of metagenomics over other methods? So basically we can identify the microorganisms, and we can also predict the functional profile of the microbial community. So which is nothing but the metabolic potential of different organisms present in the microbial community can be identified. So one limitation can be overcome in the metagenomic based approaches. So basically in PCR based amplification, so if the primer is not appropriately designed, then there are chances that certain taxa are missed. Taxa in the sense species. Certain species may be missed because of inappropriate design of primers. So this is a disadvantage with the other methods which are used for the microbial community identification. But in the case of metagenomics, since we are directly sequencing the genomes, this bias will be eliminated. So that is an advantage in the metagenomic based approach. Yes. So any doubt in the metagenomic based approaches, like how are you clear with the concepts? How is it working? Metagenomics, how metagenomics is working, and what are the advantages? Are you clear with the concepts? Hello. So what are the limitations? We have discussed about the advantages of metagenomics. So what about the limitations of metagenomics? So as we discussed earlier, metagenomics is non-targeted. We'll be completely sequencing all the genomes. So there is a possibility that we can miss the specific gene in whole genome sequence. Since we are generally sequencing all the sequences, we may miss one or two genes in the process. So sequencing depth is a limitation in metagenomics comparatively to the other methods, and higher costs. So financial and computational resources are needed to carry out metagenomics, and it is comparatively higher than the other methods. So this limits the application of metagenomics. So despite these limitations, still metagenomics has its own place in the microbial community analysis due to its potential to identify a wide range of organisms in the microbial communities. So what is metatranscriptomics? So in metagenomics specifically will be sequencing the DNA, and in the case of metatranscriptomics will be targeting RNA. So that is the major difference between the metagenomics and metatranscriptomics. So basically metatranscriptomics will give you a picture about the functional and metabolic activities of a specific community. So whereas in the metagenomics we'll be analyzing the DNA and we won't be targeting any specific genes. But in the case of metatranscriptomics, we'll be targeting RNA. so which helps us in the identification of functional and metabolic activities of the microbial community. So, so with the help of metatranscriptomics, we can get the functional insights of a microbial community. So we can also identify the environmental response of a particular microbial community. So for say if you are changing the environment, microbial environment, and with respect to the change in environment, the metabolism of microbes will also change due to the interaction of abiotic factors with the organisms. So with the help of metatranscriptomics, we can analyze those changes. What are the metabolic changes happening if we change the microenvironment where the microbial communities are present. So that is on application, and it can also be so like other omic based approaches like proteomics, metabolomics can also offer deeper insights. So as we are specifically targeting on the functional proteins and metabolites, it can also give deeper insights about the microbial community. So basically we'll be using these tools like metagenomics and metatranscriptomics to identify the functions and the capabilities of microorganisms in a microbial community. So with the help of these tools, basically we can apply these tools in bioremediation. So how we can apply these tools in the bioremediation? Basically these tools will help us to identify the microbial community and their potential. So if we know a certain microorganism can degrade a target pollutant, then I can use that microorganism for my bioremediation purposes. So this is how these tools are helpful in bioremediation. Now let us discuss how what is bioremediation and how is it working. So basically bioremediation is a controlled or natural process. It can be controlled or a natural process. So in a controlled process, we will be optimizing all the conditions for the microbes to grow, whereas in the natural process, the remediation process will happen at the natural environment. Basically biological catalysts, which are nothing but enzymes. Enzymes are will act upon the pollutants, and these biological catalysts will be utilized to remediate a wide range of environmental matrices like water, surface water, wastewater, such soil aquifers or gas streams. So this is what the term bioremediation refers to. And let us see how the bioremediation field has been developed so far. So basically the bioremediation, officially we can say the bioremediation started from 1900. So in which microbes are used in the municipal wastewater treatment. So then gradually by 1950, the microbes were started to employed in the industrial wastewater treatment process. So initially the treatment processes were limited to the municipal wastewater treatment. Then it has been extended to industrial wastewater treatment process also. So then again these bioremediation techniques were started to grow, and they were used in gasoline, which is nothing but petroleum contaminated sites. So then after gradually by 1980, people started to realize the importance of biogeochemical processes in the bioremediation. So how this biogeochemical processes are influencing bioremediation? So that insight gave a major breakthrough in the understanding the bioremediation processes. So basically in 1970s, microbial infallibility hypothesis was proposed. So in which in the hypothesis, it was proposed that microbes have their own ability to detoxify contaminants. And if we observe the technological progress in 1990, the biological processes were augmented with chemical processes for pollutant treatment. So that is a technological advancement in the bioremediation. And in 2000, in situ bioremediation was widely accepted. So we'll discuss what is in situ bioremediation in the upcoming slides. So these are the major developments that happened during the 19th century in the bioremediation. So basically, the term bioremediation is an umbrella term. It refers to both plant and microorganism based bioremediation. But microorganisms are employed to a greater extent. We'll study the reason why microorganisms are being employed instead of plants and other living organisms. So we'll be studying that also. So in bioremediation, the effectiveness of the bioremediation can also vary with respect to the physical chemical environment. So it varies with respect to the specific microbial communities and based on their metabolic potential, and also it is affected by the nature of contaminants. So the bioremediation process is affected by all these factors. And bioremediation includes all these technologies: transformation, immobilization, precipitation, elimination. So what is meant by transformation? So in the process of transformation, your contaminants will be converted into non-toxic form, whereas in the case of immobilization, the contaminants will be immobilized into the microbial cells, and in the case of precipitation, the contaminants will be allowed to settle with the help of chemicals secreted by the microorganisms, and elimination, the contaminants will be degraded. So these are the 4 different types of bioremediation techniques or approaches which are utilized by microbes to remove xenobiotics or contaminants. So what is the need for bioremediation? So basically, we not to mention the human activities have greatly affected the environment. So with approximately 39 to 50% of land is contaminated, and also in terms of atmospheric contamination, the CO2 levels have risen up to 40% over the past 140 years. This is primarily due to the hydrocarbon consumption and deforestation. So basically, hydrocarbon combustion will lead to increased CO2 emissions, and deforestation. Due to deforestation, the amount of trees available in order to capture CO2 will be reduced. So this will ultimately cause global warming. This causing global warming. And also the wide range of water matrices like groundwater aquifers, soils, rivers and oceans are being affected due to the anthropogenic activities. So that we will be discussing how we can address with the help of bioremediation and what are the basic principles driving bioremediation. So that we will be studying in the upcoming slides. So basically, contamination can be classified into inorganic and organic contamination. So in inorganic contamination, heavy metals play a major role. So they act as a major contaminants in the inorganic contamination. And in organic contamination, a wide range of organic compounds like hydrocarbons, polycyclic aromatic hydrocarbons, benzene derivatives and halogenated hydrocarbons are being served as like organic contaminants. So all these organic contaminants are arising from the anthropogenic activities. So specifically, agricultural pollutants like DDT, atrazine, and pentachlorophenol. These are like pesticides, and these contaminants can contribute to the contamination of land and water contamination. And other industrial pollutants like different solvents, dichloroethane, polychlorinated biphenyls can act as the contaminants. And other explosives can also can act as water contaminant or land contaminant. So recently, the term emerging contaminants is being used to represent a wide range of contaminants. So basically, these contaminants are present in the environment for a long time, but the effect of these contaminants are being identified only recently. So these kind of contaminants are termed as emerging contaminants, and these emerging contaminants can be classified based on the sources. Basically, these emerging contaminants can be classified as pharmaceuticals, so which are used for both human and veterinary applications. So these contaminants include antibiotics, antiparasitic agents, inophores, different anti-inflammatory drugs, anti-diabetic, anti-epileptic. So almost all the pharmaceuticals we use in our day-to-day life. And along with that, different industrial and household wastewater products also um, serve as a source of the emerging contaminants, which includes different insecticides, plasticizers, detergents, flame retardants. So all these compounds will be using in our day-to-day life, and these compounds can also serve as the source of contamination in land and water contamination. And personal care products, nanomaterials, and these compounds can also serve as the source of contamination. Basically, insect repellents. Yes, these are the common sources of emerging contaminants. Basically, commonly used to pharmaceuticals, personal care products, and other industrial chemicals are being identified as emerging contaminants. So in order to remediate these emerging contaminants, so why we should rely on bioremediation? So there are different other processes out there which are of physical and chemical nature. Then why we should rely on bioremediation? Actually, different physical chemical processes are faster than the bioremediation. Then why we should rely on bioremediation? Basically, versatility. Versatility in the sense the microbes can handle a wide range of contaminants. Um, so why we should rely on bioremediation? So basically, versatility of the process. Because of versatility of the process, So microbes can handle a wide range of contaminants, and also there will be generation of secondary waste. So in the case of other physicochemical process like absorption or any other physicochemical process, we will be needing to add external chemicals. So in that case, secondary waste generation will be there, and also the in the case of for say in the case of absorption, if you are absorbing some pollutants using an absorber, and the absorbed pollutant will stay in the absorbent, so we cannot directly dispose it and it will become a secondary waste. But in the case of bioremediation, we can directly utilize the microbes, and the microbes will degrade the contaminants, degrade or it will accumulate the contaminants, and there won't be any secondary waste generation will be there in the case of bioremediation. And it is also effective for dilute wastes. For say if we are treating a large amount of volume of wastewater where the contaminants are present in dilute concentrations, the final waste products after the treatment obtained will be acceptable in the case of bioremediation. And also if we go for any other treatment processes in the case of dilute wastes, there won't be the efficiency will be comparatively lesser than the bioremediation process due to the mass transfer limitations and the energy requirements. These dilute wastes are hard to treat with the other treatment technologies, so that is an advantage of bioremediation process. And bioremediation is also cost effective. Cost effective in the sense we don't need to add any external chemicals, and the energy requirement is also comparatively very less with respect to other processes. So these are the basic advantages of bioremediation. So and these are the reasons why we should rely on bioremediation. So in the bioremediation, why we specifically choose microbes while plants and other insects are also there? Why we should specifically go with the microbes? Because of the characteristics of the microbes. So microbes are the earliest life forms, and they have a better genetic machinery which can adapt to a wide range of environment. So that is the main reason for adopting the microbes. And these microbes are ubiquitous in nature. They can be found anywhere in the environment, and they are abundant. So they are ubiquitous and abundant and metabolically versatile, in the sense they can process a wide range of contaminants simply by altering the metabolic pathways. So these microorganisms are basically metabolically versatile. So that is an important characteristic of microbes which helps in the bioremediation. Then genomic diversity. They have a wide range of genomes which can help to degrade the contaminants, different wide range of contaminants. So genomic diversity is a unique characteristic of microbes. And small genome size. So since microbes have small genome size, and simplicity of the cell structures. So basically these microbes have small genome size, so that we can easily sequence the genome and analyze the metabolic capabilities of the microbes. And these microbes are also very simple structures, so which makes the understanding of these microbes easier, and they also have short replication times. And these are the characteristics of microbes, which gives a upper hand for microbes to be employed in the remediation processes, bioremediation processes. So this is a general representation of microbial remediation. So microbial remediation process can be classified into biotransformation, biodegradation, bioaccumulation, sorption, or precipitation. So each and every processes are completely different from each other. For say in the case of biotransformation, the pollutants will be transformed from the toxic form to non-toxic form. So basically what happens here is basically metals cannot be degraded, so they can only be converted into non-toxic forms. So in the case of chromium, chromium 6 is a toxic form, whereas chromium 4 is less toxic than the chromium 6. So in the case of biotransformation, if you are considering chromium, this chromium will be converted into toxic chromium 6 form will be converted into non-toxic chromium 4 form. So basically what happens here is the metal is reduced into its non-toxic form, because chromium 6 plus is being reduced to chromium 4 plus by electron transfer from the microbes. So this is one example of biotransformation. Usually biotransformation happens when the pollutants are not degradable. Specifically in the case of heavy metals, biotransformation is very helpful, and biodegradation happens in the case of organic compounds. Basically these organic compounds will be converted into non-toxic forms like carbon dioxide and what happens in the bioaccumulation or sorption or bioprecipitation. Basically in bioaccumulation, the pollutants will be accumulated inside the cells of microbes, inside the cell of microbes, and this can be termed as bioaccumulation, biosorption, or bioprecipitation. So mostly metals will undergo these process of biosorption or bioaccumulation or bioprecipitation. So what is the basis behind microbial remediation? So basically these microbes consider pollutants as the resources, and in the case of organic compounds, these organic compounds will serve as sources of carbon, and they can also serve as the electron donor. Whereas these heavy metals are also serving as the electron donors or acceptors or even nutrients, while other inorganics like nitrate, phosphate and sulfate, these other inorganic compounds can also act as electron donors or acceptors. So how detoxification is happening in the case of radiation? So basically pollutants will be either transformed or being used as a source of energy, and in this case co-metabolism happens. Basically your pollutants won't be the primary source of energy in the microbes, and it serves as the additional energy source. So this is the basis behind microbial remediation. So how these organic and inorganic pollutants are processed by different organisms? So the organisms can be classified into different categories based on their nutrient utilization ability or mode of nutrition. So basically, organisms can be classified as phototrophs, autotrophs, chemotrophs, and heterotrophs. And these phototrophs can be classified as into photoautotrophs, photoheterotrophs, and these phototrophs basically use the light energy to generate ATP and PMF, which are sources of which are energy reserves. In the case of autotrophs, specifically in the case of autotrophs, either light energy or chemical energy can be utilized to generate the energy required to carry out the metabolic processes. Whereas in the case of chemotrophs can be classified into chemoheterotrophs and chemoautotrophs. And in the case of chemoautotrophs, they don't need to rely on any external factors to generate their own food. Whereas in the case of chemoheterotrophs, they have to rely on the external sources to generate their own food. And heterotrophs, and these heterotrophs can be classified as chemoheterotrophs and photoheterotrophs. So, despite the differences in their mode of nutrition, almost all the organisms can use the organic carbon sources and also the nutrients, other inorganic nutrients. So accordingly, the metabolic pathways residing in the microbial cells will be altered in order to utilize the organic and inorganic sources present in the surroundings. So this is how microbial remediation is working. So what are the requirements for microbial remediation to happen? So microorganisms are the primary requirement, and these microorganisms need an energy source and the electron acceptors. Then other environmental conditions like moisture, pH, nutrients, and temperature are required. And finally, there shouldn't be any toxic substances to inhibit the microbial activity. And there should not be any predators which inhibit the microbial activity. And these are the basic requirements that are needed to carry out the bioremediation process. So what are the advantages of bioremediation? So bioremediation can be used to directly remediate the contaminated site. So we can clean the wastes in situ, and organic wastes can be completely destructed and can be converted into non-toxic forms like water, carbon dioxide, and other mineral salts. And it is a natural process. And minimal environmental disruption, since we are not using any external substances or toxic substances to carry out the remediation processes. It leads to minimal environmental disruption. And these bioremediation processes is effective in the case of hydrophobic pollutants, which are hard to remove in the case of other treatment processes. And these processes are environmentally friendly. And there is no need for dewatering. In the sense, so if the aquifers are contaminated with some contaminants, So if we want to treat them, we can directly employ the microbes there. So there is no need for dewatering and treating the environment. So that is one advantage, and we can integrate other technologies with the microbial remediation. So these are the basic advantages of bioremediation comparing to other processes. And this bioremediation process has also its own disadvantages. In the case of heavy metals, biological processes are not so efficient due to their toxicness. And also in the case of heavy metals, the heavy metals will only be converted into non-toxic forms and will not be completely eliminated from the environment. So that is some disadvantage. And we need to monitor the process continuously. That is a disadvantage of the bioremediation process. Sometimes if some predators or contamination happens in the among the microbial community, that will also affect the process effectiveness. So we have to continuously monitor the process, and the success rate will vary depending on the environmental factors. And at high concentration of pollutants, the microbial activity can be inhibited due to substrate inhibition. So that is one limitation. And scientifically intensive, which means before identifying the microbial community, possible microbial community, we have to undergo a lot of processes like screening, genome analysis, identification, and a lot of processes are all involved in the framing a bioremediation technique. So it is scientifically intensive to implement a bioremediation technique in the contaminated sites. And there is also a potential risk of toxic byproduct accumulation. For say, if a microbe cannot completely degrade a xenobiotic, and the degraded product is also toxic, then that is a disadvantage. So these are the major limitations of bioremediation. So basically, bioremediation can be classified into in situ bioremediation and ex situ bioremediation. In in situ bioremediation, contaminants will be treated at the site of contamination itself, whereas in the ex situ bioremediation, the contaminants will be excavated and transported to the treatment systems. Other than that, again, this bioremediation processes can be classified into engineered and intrinsic processes. In engineered bioremediation process, so different engineering techniques will be used to enhance the bioremediation process. And in intrinsic bioremediation, we won't be using any external entities to enhance the bioremediation process. Simply it will happen, the bioremediation process will happen on its own. So these are the types of bioremediation processes based on the site of remediation and also the like sophistication of the process. And bioremediation approaches can be and bioremediation approaches can be can be a biostimulative approach or a bioaugmentative approach. So basically in biostimulation what happens is the activity of resident bacteria will be stimulated in order to remediate the target chemicals by adding specific nutrients, water, electron donors, etc., and other substances. So this is termed as biostimulation, so in which we will be stimulating the microbial activity, and augmentation. So basically we will be employing specific organism which has the degradation ability to break down the contaminants. So in the case of biostimulation, it will stimulate the activity of microbe, even though the microbe is not having the ability to degrade a particular contaminant. With the biostimulation approach, we can make that organism to degrade the target contaminant. Whereas in the case of augmentation, we'll be utilizing the specific organism which has the inherent ability to degrade the contaminants. So this is the difference, basic difference between these two approaches, biostimulation and bioaugmentation. So how this bioremediation approaches should be framed? Basically that microorganisms should be present in the contaminated environment. So then only it will have the ability to remediate the contaminated environment. So that is the basic, one of the basic criteria. And their metabolic capability should be well understood, so that we can say that whether it has the ability to degrade the contaminants. And their responses to the environmental condition, how the microbe is behaving to the environmental condition. So all these factors determine the bioremediating capability of a microbe. So before undergoing genomic approaches, we were relying on the non-molecular techniques like simple isolation and characterization. So in order to identify the possible microorganisms to degrade the contaminants, and these processes cannot be used for organisms which cannot be grown in the laboratory scale setups. So basically in the pre-genomic approaches, we can isolate and characterize the responsible organism for carrying out the degradation process. And this approach is limited to organisms which can only be grown in the laboratory scale settings. And this non-molecular based techniques basically helps to identify the potential amendments that can be used for engineered bioremediation. And these processes are only limited to organisms which can be grown in the laboratory scale setups. So what are the applications, how can we apply the bioremediation process? So what are the steps involved? So initially we should isolate, amplify and apply the organism, which means we should isolate the organisms which are capable of degrading the contaminants, and then we should amplify it, in the sense we should use some enrichment cultures to amplify the population of the organisms which can degrade the contaminants. Finally, we should apply the organisms for remediation. Then we should create a functional proof, which means we have to prove that a particular organism is having the capacity to involve in the degradation. So that functional proof we have to provide. And high biomass protection is also required in order to increase the process efficiency. And simple surface remediation in the sense we can apply these processes in the local surfaces also for directly cleaning up the contaminant. And niche applications in the sense we can apply the microbes in the liquid stream directly. So this is how bioremediation processes are working. And they also has the certain limitations. So there are only small amount of species that can be cultured, for about 0.1 to 10% of species. So only these species can be utilized in the process of bioremediation, because only if we can culture a species, they can be studied well. Anyway, different advanced techniques are there without culturing the organisms can also be can study, but these culturable organisms can be best studied among the other organisms. And there will be in situ competition among the organisms, which will also reduce the process efficiency. So these are the basic limitations in the bioremediation processes, application of bioremediation. So let's have a brief recap of what we have learned today. So initially we learned about the metagenomic approaches. So basically in metagenomics we'll be studying the all the genomes present in an environment by directly sequencing it. Let's have a brief recap of today's session. So initially we learned about the concepts of metagenomics. So basically metagenomics can be considered as an alternative to PCR based techniques in order to study the microbial community. Since in metagenomics the entire genome will be directly sequenced, it has some advantages over the other methods, since all the whole genome will be sequenced without missing any taxon. And with the help of metagenomics, we can identify the community composition, and we can study the metabolic potential of different genes and pathways. And we can construct models based on the metagenomic data. So these are the basic outcomes of metagenomics. And metagenomics has several advantages over other methods. So basically metagenomics helps to functional profiling of microbial community, and it also avoids biases that are caused due to PCR amplification. So first, if primer is not designed for a particular taxon, then the chances are there that particular taxon will be missed. So basically high precision is an advantage in the metagenomic approaches. And metagenomics has also its own disadvantages, like it has a comparatively lower sequencing depth, and cost of carrying out metagenomic studies is higher due to the requirement of computational sources and financial requirements. But still, it has a great potential to analyze the microbial community and to study the microbial ecology. So metatranscriptomics is an alternative to metagenomics, in which we'll be relying on RNA-based studies instead of DNA. Through RNA, studying the RNA, we can get the functional insights. So in other than metatranscriptomics, other approaches like metaproteomics, metabolomics can also be considered as an alternative for the metagenomics. And this omic-based approaches will help in understanding the metabolism and also the response of different microbial community towards the environmental conditions. So these using these tools, we can study the microbial community, and finally we can apply the microbes like microbes which has the ability to degrade the contaminants, environmental contaminants. So coming to the bioremediation, this bioremediation is a umbrella term which is used to denote remediation of different environmental matrices like water, surface water, wastewater, sludge, soil, aquifers, or other gas streams. So on this in bioremediation, the advancement happened almost in the 19th century. So at 1900s, first the bioremediation was applied in the municipal wastewater treatment. Then gradually by 1950, the bioremediation process was also used for industrial wastewater treatment. Then by 1970, the different microbes were used to remediate the gasoline contaminated sites. And in 1980, the biogeochemical processes were gained attention. And theoretically, the microbial infallibility theory was developed in the 1970s, which proposes that the microorganisms have their own inherent ability to detoxify contaminants. And the great technological advancements happened in the 1990s and in the 2000s, where the hybrid processes were developed, where the biological and other chemical-based approaches were augmented together to remediate the contaminated sites. And in the 2000s, in situ bioremediation was widely accepted. And these bioremediation processes can be basically classified into 4 types: transformation, immobilization, precipitation, and elimination. In transformation, the micro will transform the contaminants from one form to another form, which is non-toxic. And in the case of immobilization, the contaminants will be immobilized inside the cells. And in the case of precipitation, the same happens. The contaminants will be precipitated with the help of chemicals secreted by the microbes. And in the case of elimination, the contaminants will be degraded. So what is the need for bioremediation? So basically, our environment is heavily contaminated, both in the terms of atmospheric contamination and also the land and water contamination. So that comes the urgent need for the bioremediation processes. And this bioremediation process can be utilized to remove a wide range of contaminants like inorganic and organic contaminants. And these contaminants can be classified based on the chemical nature and also the sources. And the term emerging contaminants has been coined to mention contaminants which are present in the environment for a long time, but their adverse effects were identified only recently. So these contaminants are termed as emerging contaminants. So these emerging contaminants can be classified based on their sources like pharmaceuticals and personal care products, industrial chemicals, etc. So why we should rely on bioremediation? Because of the versatility of the process, stability of the process in which the contaminants will be, a wide range of contaminants can be degraded with the help of microbes, and no secondary waste will be generated. um, and the bioremediation process is also effective for a daily basis, and it is also cost effective. Why we should rely on microbes when other organisms are also present? Because these are like earliest microbes are earliest like forms, so which has the genetic machinery which can adapt to any kind of environment, and these microbes are ubiquitous and abundant, metabolically versatile, and they are dominant, and it has a small genome size, which makes it easier for the researchers to study completely, and it also has a simple cell structure and short replication times. So all these features makes the microbial remediation a more favorable process compared to the other process. And the microbial remediation can be classified into biotransformation, biodegradation, and bioaccumulation, or biosorption, or bioprecipitation. So basically, in biotransformation, the contaminants will be converted into toxic form to non-toxic form, and in the case of biodegradation, the organic compounds will be converted into non-toxic CO2 and H2O. Whereas in the case of bioaccumulation, the contaminants will be accumulated into the cell or precipitated into the cell. So basically, this organic compound serves as the carbon and the electron source, serves as the carbon source and also the electron donors. Whereas the heavy metals serves as the electron donors and acceptors, or even nutrients, and other inorganics like nitrogen, phosphorus, and sulfur serves as the nutrients and electron donors or acceptors. So how these pollutants are degraded? So actually, the microbes will undergo co-metabolism, in which the pollutants will be considered as a secondary source of energy, and it will be utilized by the microorganisms. This is how bioremediation works. For bioremediation, basically we need the microbes to carry out the remediation processes, and we need energy sources and electron acceptors for the microbes, and we need to set up the necessary environmental condition. Then finally, there should not be presence of toxicity and predators, and metabolites should be completely removed from the system. So these are the basic requirements for bioremediation processes. So what are the advantages? So bioremediation helps in the in situ cleanup. The bioremediation process can be directly applied in the contaminated site, and organic wastes can be completely disrupted into inorganic forms, which are non-toxic, and these processes are natural and won't cause any environmental perturbances. And also, these processes are more effective in the case of hydrophobic contaminants, which are hard to remove with the other processes, and these processes are environmentally friendly. And these bioremediation processes can be integrated with the other technologies. So it also has its own disadvantage. So heavy metals can only be converted into non-toxic forms and will not be completely eliminated, and it needs extensive monitoring, and the success rate will vary according to the environmental condition. At high concentration of pollutant, inhibition happens, and the process is also scientifically intensive, which means we need a substantial scientific expertise to understand the microorganism and to employ them in the environmental processes. And toxic byproduct generation is also a risk in the case of recalcitrant contaminants. The bioremediation processes can be classified based on the site of remediation. So the bioremediation can be classified as in situ and ex situ remediation. In situ remediation, the treatment can happen at the site of contamination itself, whereas in the ex situ remediation, the contaminants will be transferred to the treatment site from the contaminated site. And again, the bioremediation process can be classified as engineered and intrinsic processes. And in engineered bioremediation, the microbes will be stimulated with the scientific approaches, whereas in the case of intrinsic bioremediation, the degradation or the removal of contaminants happens with the natural rates of substrate and nutrient supply, and there won't be any uh, influence of uh, engineered processes. So this is how the bioremediation process can be classified. And bioremediation approach can be classified as biostimulation and bioaugmentation. In the case of biostimulation, we will be stimulating the microbial activity by adding nutrients, water, and electron donors and acceptors and other substances. In the case of bioaugmentation, the microbes will be directly employed in the degradation or the removal of contaminants, which has the inherent ability to remove the contaminants. So bioremediation can be applied based on the following principles. So basically, we should isolate the organism of interest which can degrade the contaminants, target contaminant, and we should amplify it, and finally we have to apply the contaminated set. On functional proof, we have to create a functional proof in the sense we have to demonstrate the organism's capacity to involve in the degradation process, and we should also ensure the high biomass production so that the efficiency of contaminant removal will be maintained. And this bioremediation process can be applied in the simple surface level remediation, which means local surface can be remediated with the help of microbes by employing directly on the contaminated side, employing directly the microbes on the contaminated side. So, but the bioremediation process has also their own limitations. Only culturable species can be employed in the bioremediation processes, and there will also be competition among the organisms, which will reduce the ultimately reduce the ability of the microorganisms to involve in the remediation. Yeah, thank you. So if there are no doubts, then we can wrap up the session. Yeah, thank you, Varun.